Welcome back to the Deep Dive. Today, we are strapping in for kind of a molecular speed run, really. We're looking deep into the foundational chemistry of life itself. We've got a dense stack of sources here, all focused entirely on enzymes. If you've ever just stopped and thought about the sheer, metabolic chaos happening inside your body,
Mm.
thousands of complex reactions every single second, you kind of have to ask, how does it all happen so fast? How do we manage all that without, well, constantly exploding or just seizing up?
Yeah, it's incredible. And the answer is we've got this invisible, highly structured management team running the show. Enzymes, they are the essential speed regulators. So our mission today really is to define their core job, understand the honestly brilliant mechanics of how they operate, and maybe most importantly, figure out what makes these really powerful but also delicate machines break down.
Okay, let's jump straight to that core definition then, because this feels like where the whole deep dive pivots, right? We know they're fundamental, but structurally, what are we actually talking about?
We're talking about large proteins, and they're defined by their incredibly complex three-dimensional tertiary structure. That shape is absolutely critical, non-negotiable, but their function is what makes them so unique. They are biological catalysts.
Biological catalysts.
So
Okay.
a catalyst is just a chemical that speeds up a reaction, but it doesn't get consumed itself. An enzyme does exactly that, but within a living system. It's like this reusable workhorse that can do the same job millions of times.
Right. And the mechanism behind that speed, that seems like the most critical takeaway for anyone learning this, doesn't it? It's not just that they rush things along. They fundamentally change what's needed for the reaction to even happen.
Exactly. You can think of them as energy managers. Every single chemical reaction needs a kind of kickstart, a minimum amount of energy just to get going. We call this the energy of activation. Now, without an enzyme, that energy requirement might be huge. The reaction could take like years to happen at normal body temperature.
So the enzyme basically just lowers that barrier and it makes it easier to get started.
Precisely. Imagine you need to get a tiny toy car over a massive mountain to reach the finish line. That mountain is the activation energy. The enzyme doesn't give the car more power or anything. Instead, it provides this super efficient shortcut, like a tunnel straight through the base of the mountain. You need way less energy to push the car through that tunnel than to heave it over the whole mountain, right?
Right.
That reduction in the required energy, that is the key to biological speed.
Okay, that makes sense. So let's look at the versatility of these tunnel providers. Once the enzyme and its target molecule, the substrate you called it, once they get together, what kind of work can they actually do?
Well, our sources highlight this really vital dual function. It's necessary for pretty much all of metabolism. Depending on the specific enzyme's job, it might act like a construction crew or maybe a demolition crew.
Okay.
So they can either join substrate molecules together, build larger complex compounds, or they can break them apart, which is essential for things like digestion or recycling materials in the cell.
That dual function is pretty impressive, building and breaking. But the moment they interact, how the enzyme and the substrate actually recognize each other, I understand that our understanding of that has actually changed quite a bit over time. What did scientists first think this looked like?
Yeah, it's a fascinating story. For a very long time, the main theory was called the lock and key model. It was a beautifully simple idea, really, but ultimately maybe too rigid.
Bridget, how?
Well, the enzyme, the lock, was thought to have this fixed, completely unchangeable shape at its active site. That's the spot where the work happens. And the substrate, the key, it had to match that shape perfectly, exactly, right from the start. The key didn't fit, bang, the reaction just couldn't happen. It really emphasized this perfect static specificity.
But the body's rarely static, is it? Things are usually more flexible.
Exactly, and that's where the modern understanding comes in. This is where it gets, I think, really interesting. We now use what's called the induced fit model. This model acknowledges that the active site isn't some rigid, carved piece of wood. It's actually flexible. It can change shape slightly.
Okay, how does that work?
Well, when the substrate binds to the enzyme, the binding process itself actually causes or induces the enzyme to subtly alter or refine its shape. Think of it like putting your hand into a glove that's maybe just a tiny bit loose.
Right.
The moment your hand is fully in, the glove subtly molds and adjusts to the contours of your fingers, right? It creates a much tighter, much more effective grip.
Ah, I see. So the fit gets better after they connect.
Exactly. A better grip ensures optimal catalysis and makes the whole process much more efficient.
That dynamic responsiveness, that feels like a huge leap in understanding. It means the body's machinery isn't just pre-built. It's constantly sort of negotiating and refining the fit.
It really speaks to the efficiency of biological systems. Yeah, it allows the enzyme not just to bind the substrate, but also to put maximum chemical stress on the specific bonds that need to be broken or formed. Very clever.
Okay, but if this mechanism is so perfectly refined and responsive, what's the weak spot? What makes these highly specialized systems break down? Because even a slight malfunction sounds like it could lead to metabolic disaster.
You've hit on the key vulnerability there. The environment matters.
Yeah.
Profoundly, because the enzyme's function relies entirely on that complex tertiary structure, that specific 3D shape. We talk about anything that messes with the shape messes with the function.
Okay, like what?
Well, let's start with temperature. Our sources really emphasize that the optimal window for human enzymes is pretty narrow. It centers right around our average body temperature, 37 degrees Celsius.
Right. But we know fevers happen, people get hot. What's the consequence if the system gets too hot?
Yeah, if the temperature climbs above 40 degrees Celsius, that's about 104 Fahrenheit, the enzyme will stop functioning, potentially permanently. It's because that high temperature provides too much energy. The delicate protein structure starts to vibrate too violently, and it basically unravels. It loses its shape. We call this process denaturation.
Wait, denaturation. So when we talk about a high fever being dangerous, especially for, say, brain function,
Yeah.
is that what's happening? A direct result of enzyme denaturation. Are we literally breaking the machinery?
That is precisely the connection, yes. If those key metabolic enzymes, the ones running those tunnel shortcuts, if they denature, the reactions slow way down or they stop entirely. The body's whole speed governance mechanism just collapses. It's actually the same reason cooking food works, you know? The heat denatures the enzymes in the food, changes its texture, makes it easier for us to digest.
Wow. Okay, that makes the danger zone incredibly clear. And presumably, pH is just as crucial, right? But it's not just the overall body pH that matters. It's the specific optimal pH for each unique enzyme. Is that right?
Exactly right. Unlike temperature, which can be kind of a global issue for the body, pH is often very localized. So an enzyme working in the super acidic environment of your stomach, like pepsin, it works best at a pH around 2. Very acidic. But an enzyme in your small intestine needs a near neutral or even slightly alkaline environment to work properly.
Big difference.
Huge difference. And if you take an enzyme and shift it outside its required narrow pH range, well, the change in hydrogen ion concentration interferes with the bonds holding that tertiary structure together, and the result, denaturation. Same as with too much heat. The enzyme loses its shape and becomes inactive.
Okay, so environmental threats,
Yeah.
temperature and pH.
Got it.
But let's move on to capacity constraints. This is where concentration comes in, right? The rate of the reaction must be some kind of balancing act between how much catalyst you have and how much raw material.
Yeah, that leads us nicely to the saturation principle. Let's first look at the enzymes themselves. If we increase the enzyme concentration in a system, we're essentially providing more workers, more active sites, for the substrates to use. So naturally, the reaction rate speeds up, quite dramatically usually.
Okay.
There's a limit, I assume. That benefit only lasts until you run out of inputs.
Correct. The reaction rate will eventually plateau. It levels off because all the available raw material, the substrate, has been used up, or at least is being processed as fast as possible. At that point, adding more enzymes is pointless. You don't need more workers if there's no work to do. You'd need to add more substrate to push the rate higher again.
Okay. And the reverse is also true then for substrate concentration. If we increase the amount of substrate.
Yep.
Initially, the reaction speeds up because more substrate molecules are bumping into and binding with the available active sites.
Makes sense.
But eventually you reach saturation again. Think of it like rush hour traffic on a 4-lane highway. You can add more cars, that's the substrate, but once all 4 lanes, those are the active sites, are completely full, the rate is maxed out. Traffic can't go any faster. If you add even more substrate, more cars, the overall rate won't increase because the system is limited by the number of available lanes, the number of working enzymes.
That concept of saturation seems absolutely vital.
Yeah.
Especially when you think about things like, I don't know, drug delivery or maybe metabolic disorders. If a medication works by binding to an enzyme, you can only push the dosage so far before you just hit that limit, and the body literally can't process it any faster.
Exactly. It determines the maximum velocity of that metabolic pathway. Vmax, we call it sometimes. Once those active sites are full, the only way to increase the overall rate is to actually synthesize more of the enzyme itself, build more lanes on the highway.
Fascinating. Okay. So enzymes are reused. They're these resilient workhorses. But you mentioned that delicate tertiary structure holds the key. And to hold that shape and do its complex job, the enzyme often needs a bit of help, right? It needs a support crew. Which brings us, I think, to coenzymes and cofactors.
Right. If the enzyme is the sophisticated engine that drives the reaction, then the coenzymes and cofactors are like the essential fluids and parts the oil, the spark plugs, maybe the transmission fluid. They ensure the engine can actually turn over and hold together properly. Without them, the protein might be there, but it remains incomplete or inactive. It can't do its job.
Okay, and our sources draw a pretty clear chemical distinction between these two helpers. Cofactors are defined as.
Cofactors are typically inorganic compounds. So in the context of nutrition, this means we are primarily talking about minerals, things like magnesium, iron, zinc,
Got it. Minerals.
Oh.
And coenzymes then are mainly defined
as organic compounds, which for us, nutrition-wise, means we are talking about vitamins. These vitamins are absolutely crucial for metabolism because they often act as carriers or facilitators. They might literally help the active site grab onto or manipulate the substrate during the reaction.
Wow. So your daily diet isn't just about getting fuel for energy. It's about supplying the specific, sophisticated molecular tools your enzyme machinery actually requires to function.
That's a great way to put it.
Let's look at the vitamins first, then the organic coenzymes. There's a crucial distinction based on solubility, right? How the body handles them.
Yes, absolutely. We basically classify vitamins by how the body stores or excretes them. First, you have the water-soluble vitamins. These are the B vitamins and vitamin C. The key thing here is they are not stored in the body in any significant amount. They are essential coenzymes for tons of processes like energy production, making neurotransmitters, collagen synthesis. But they generally need to be replenished daily. Any excess you take in just gets flushed out, usually in urine.
That's a huge nutritional difference compared to the other group, the fat-soluble ones.
It really is. The fat-soluble vitamins, we're talking A, D, E, and K here, are stored by the body, primarily in the liver and in fat tissue. Because the body can hold on to them for extended periods, you don't necessarily need to consume them every single day. There's a reserve.
Okay, let's make that connection even more real for the listener. If we look at a deficiency, maybe vitamin C, a water-soluble coenzyme. You mentioned collagen synthesis. That's its main coenzyme job.
One of its major ones, yes. It's crucial for the enzymes that build and maintain healthy collagen, which is the protein that basically holds our tissues together, skin, blood vessels, gums.
Okay, so what happens if that coenzyme, the vitamin C, is missing? Because it's water-soluble, not stored.
Well, the machinery stalls. The enzyme is there, but it can't do its job without its helper. And the famous dramatic result of severe vitamin C deficiency is scurvy. The reason sailors historically got bleeding gums, wounds wouldn't heal, teeth fell out. It wasn't because their bodies stopped trying to make collagen. It was because the specific enzymes responsible for building healthy, strong collagen were robbed of their essential support crew, that vitamin C coenzyme. The machine's intact, but it just can't function without the vitamin.
That really drives the point home.
Oh.
Okay, now let's touch on the minerals, the inorganic cofactors. They're also classified, but based on the quantity we need rather than solubility.
That's right. We divide them into macrominerals, which, as the name suggests, are needed in larger amounts. Think calcium and phosphorus for bone structure, sodium and potassium for nerve function, magnesium for hundreds of different enzymatic reactions.
And those, wow.
Yeah, magnesium's a busy one. And then we have the trace minerals. These are needed in much smaller amounts, but they are absolutely no less critical. This group includes things like iron, zinc, iodine, copper, selenium.
Let's take iron as an example, a classic trace mineral.
Okay, iron. It's an essential cofactor, part of the structure for enzymes involved in carrying oxygen, like hemoglobin, but also for enzymes critical to energy production within the cells. So without adequate iron, those enzyme systems just can't work efficiently, which is why the result is fatigue, weakness, anemia.
So whether the support crew is an organic coenzyme like a vitamin or an inorganic cofactor like a mineral, their presence is just non-negotiable for keeping metabolism running at speed.
Absolutely non-negotiable.
Okay, this brings everything together really nicely, I think. We started by defining enzymes as these crucial biological catalysts, their main job, solving the energy problem by lowering the energy of activation. They are fundamentally the reason life can happen fast enough to sustain itself.
And we move through the more modern dynamic induced fit model, how the enzyme isn't just a passive lock, but an active site that actually adjusts its 3D shape for a much better, more effective interaction with its substrate, like that glove anatomy.
Right, the glove. And we saw just how fragile that whole speed mechanism is. A change in temperature above that critical 40 degrees Celsius mark or a significant deviation from the optimal pH. It causes immediate and potentially catastrophic denaturation, the loss of that crucial 3D structure, which just grinds the necessary reactions to a halt.
And finally, we established that really direct link between what's on your dinner plate and the machinery inside your cells. These resilient enzyme proteins often absolutely require coenzymes, which are mostly vitamins, and cofactors, mostly minerals, to perform at speed. It makes nutrition about far more than just getting calories. It's literally about supplying the spare parts and the support crew for your metabolism.
Yeah, that connectivity back to daily life feels paramount.
Yeah.
And it leads to a final thought, doesn't it? Since we know that key water-soluble vitamins, like the B vitamins and vitamin C, are essential coenzymes needed constantly to maintain those high metabolic speeds. And yet, as our sources emphasized, they are not stored by the body.
Right. It really raises an important question for you, the listener, to consider. Knowing that these key metabolic engines need these specific coenzymes to be present pretty much every single day, and knowing the body just gets rid of any excess, well, how incredibly fine-tuned must our daily intake of these specific organic compounds actually be? How precisely do we need to supply them to maintain the constant breakneck speed of all these essential physiological processes without any interruption?
It definitely gives you a new level of respect for that daily diet, maybe even that multivitamin, something to chew on, thinking about all the microscopic chaos that's necessary just for life to continue moment by moment.
Absolutely. Food for thought, quite literally.
We'll catch you on the next deep dive.